This podcast. So, welcome to the Knowledge Nights show. And before we proceed, I would love to introduce you, like who we have today. We have Dr. Neal Silverman, and he's a research scientist at University of Massachusetts Medical School. He has studied at UC Berkeley, MIT, and Harvard, like all the great schools to pursue his career. And he has published over 73 research papers from his lab and have been actively involved with research on Drosophila melanogaster and its innate immune system.
My pleasure. Nice to meet you both and talk with you.
So we want to talk about your profession, your journey, like mainly for people who are aspiring to go into research field and labs, and yeah, some of your life instances. So before we proceed, a quick introduction about my background and Pratham's background, who's the co-host here. So I'm currently a final year student, and I'm pursuing literature from Delhi, India. And apart from that, I've been into debating and I wish to pursue a career personally in advertising. So yeah, that's just like pretty much about me.
Literature.
Hello, Dr. Neal. I'm Pratham, and I'm a high school student studying biology, and I wish to pursue research degree further. So I'm very excited to have a talk with you. So let's get started with the first question. I really want to know deep dive into your journey and how you land up in this research field of immunology. I'd really like to know that.
Yeah. So how I ended up in immunology is a little bit accidental, but my larger journey, starting with how I got interested in life sciences, biology, goes back to when I was in high school, and I took all the science classes my high school offered, which was the usual chemistry, physics, biology. I think that was about it. And I really liked biology. It was just really fascinating. I remember learning very sort of elementary things about how DNA encoded genes and genes got transcribed into RNA, and thinking how marvelous it was and how amazing it is that you got to turn all these things, all these genes on and off at all the right times. And it just really sparked my fascination. So when I went to university, I headed off intending to study molecular biology, and I stuck with that. I'd say, in the educational system in the United States, which is maybe different from India, students often switch their majors in large degrees, in large swings. They come in thinking they're going to go to medical school and they're going to study biology or chemistry or some hard science, and they decide halfway through their freshman year that it's not for them, and they study literature and they end up being lawyers, or there's lots of changes. But I was very focused, and I stuck with my original plans of studying molecular biology. I got involved. What was key for me was getting involved in the research lab as an undergraduate. That's what I always encourage students to do when they ask me about how to get involved in research and how to become a researcher. The first thing you have to do is to find out if you actually like doing research, because laboratory research can be tough. We have experiments, we have hypotheses that we're testing, and we're trying to do experiments, and sometimes the hypotheses are just wrong. I mean, our ideas are just wrong, and you have to be ready to accept that. Other times your experiments don't work for other reasons, not because your hypothesis is wrong, but because the experimental design was not able to really address the question, or you weren't able to generate the tools or reagents that you need to do the experiments. And so it can be frustrating, and there's a lot of failure in bench science, and you spend a lot of time troubleshooting, trying to figure out why your experiment's not working the way you think it should. So it's not, people can love life science and not like actually being experimental biologists, because it can be challenging mentally mentally challenging. So I always encourage students to try and get involved in research when they go to university. When I was in university, we didn't have email, wasn't the thing that everybody used all the time. So I literally had to just go knock on some doors and make some phone calls to find myself my first research position. And I was lucky and privileged. And my first research position was a summer position in a laboratory, actually at UCLA, because I grew up in Los Angeles. And so it was one summer when I was home, and it turned out to be a friend of a friend who was a faculty member at UCLA. And I called him up, and he was happy to have me come work in the lab for the summer. And I really liked it. So although it's not for everybody, I really enjoyed it. I enjoyed, at the beginning, you just sort of learn how to do things in the laboratory. And I enjoyed, growing up plasmids and growing cells and counting plaques, doing very basic stuff. But it was just so much fun to be in the lab and trying to discover new things. And then I went back to Berkeley that term, and I knocked on some more doors, and I got a position in a lab studying developmental biology. So I was doing developmental biology with frogs, with Xenopus laevis, which is a African clawed toad frog, maybe. I don't remember what it's called anymore. And so for a couple of years there, I was a budding developmental biologist. And then I went off to, and when I was finishing university, I liked the lab, and I really liked this idea, almost romantic idea of being involved in discovering new things. That just seemed like where I wanted to be. I wasn't interested in making a lot of money. I wasn't interested in making a product. I wanted to discover things. That just seemed like, I think I was definitely a bit of a romantic. That just seemed like that's the place I wanted to be. And so that's what graduate school's for. You actually have to discover something to earn a doctorate. You have to discover something and write a thesis and about your discovery, and then defend your ideas. So I applied to graduate school. I applied to quite a few, and I decided, since I stayed on the West Coast for my university days, I decided to go to the East Coast for graduate school, and I went to MIT. I had a few good options, so I could stay on the West Coast. My parents were kind of mad at me for going back east. I thought it'd only be for like 5 or 6 years, and that was 30 years ago. I'm still here. So the program that I went to in graduate school, it was a biology program, but it wasn't focused. There was all sorts of things: developmental biology, immunology, neurobiology, protein biochemistry, a huge range of possibilities. And that's one of the reasons that particular program was at MIT appealed to me, because I didn't have to decide that I wanted to be a developmental biologist when I was 22, or I wanted to be an immunologist or a yeast geneticist. I could sample a bunch of things, take classes that first year, sample different opportunities in different labs, and decide which lab I wanted to settle on. So that's what I did. I went out there, and for my graduate work, I did something totally different from the year or so I spent trying to do developmental biology. When I was an undergrad, I worked with a kind of yeast called Saccharomyces cerevisiae. This is actually the yeast that we use to make bread and make beer. And it's a favorite molecular and genetic experimental system that's used in labs all over the world. It's very powerful. It has a haploid genome, which means it has one copy of each chromosome. At least it can't have two, because it can mate and become a diploid, but there's a haploid form. You do lots of experiments in the haploid form. Makes the genetics really simple, because you don't have to worry about 2 copies of the gene. And in graduate school, I worked on a problem about how genes get turned on and off. So genes get transcribed, so the DNA gets transcribed into RNA. And to turn a gene on, the first thing you need to do is make that RNA copy. That's called transcription. And we worked on how transcription was activated. So genes can be not transcribed or transcribed to very, very high levels. And we were trying to understand how you got that high level, how genes got turned on to these high levels. And it's a process called transcriptional activation. And it was very, especially at the beginning, it was super fun. There was a lot of things we didn't know, and we devised, I was part of a team, and we devised a forward genetic screen to look for mutants that were defective in turning genes on in general. We found some interesting genes, and then I kind of settled in trying to understand what one of those genes and the protein that it encoded, that it made, did. That was what I did for my graduate work. It was an interesting few years. Well, we tried to figure out how it worked. Our progress was slow and kind of baby steps. I think we were able to show it was really involved in something about the transcriptional process, but we didn't really understand how it did that. And we weren't thinking about chromatin at all. So DNA is wrapped in up in these tight bundles called chromatin, and the key protein in the chromatin unit is a histone. And we weren't thinking too much about the histones in the chromatin. We were thinking more about like what DNA would do when it was more naked. And another group, a group in Rochester from David Alice, working in a totally different system, right when I was finishing up my PhD, had a paper publication that kind of blew open the whole field and really changed the way we think about everything that we were doing. And in particular, they were working with a single cell eukaryotic organism called tetrahymena, which has a lot of very interesting biology. And they've been working on that in years. And they'd noticed in tetrahymena that genes that were transcribed, that were actively transcribed, the histones around that gene were acetylated. And they went on and biochemically purified a protein that acetylated the histones. And it turned out that their histone acetyltransferase, that's what that enzyme's called, was one of the genes that we had gotten in our screen. And we'd actually in our screen gotten a whole bunch of genes that form a big complex that are involved in, we know now, modifying histones, in particular acetylating histones. They do some other things to histones too, and it's this acetylating the histones that is critical for allowing genes to be transcribed. And that's a huge field now. I'm not involved in that field anymore, but that was the very early days. By the time I graduated, finished defending my thesis, I was kind of tired of transcription. It's a very competitive field. I felt abused, quite frankly. And I was looking for something else. I still like science. I still like this idea of discovering new things. And I was looking for some area to move into that was a little bit more open, a little bit more new, and certainly new to me. And at first I started thinking about getting into microbial pathogenesis. So this is how, say, bacteria or viruses, how they cause disease, how they infect cells and cause disease. In particular, like the virulence mechanisms that really make animals, people, cells sick. I started looking around in that, and quite frankly, I didn't find a good opportunity for a postdoc in that area. I was interested in staying in Boston area at that time for personal reasons, and I didn't find any opportunities that I really loved. So I branched out a little bit, and I said, well, I'm interested in this, how microbes make cells and animals so sick. And I got interested in how animals respond and animal cells respond. And of course, that's what the immune response is. And of course, pathogenesis or disease is always something about the balance or the battle between the microbe on the one hand and the host cell and the host on the other hand. And I think, if I can diverge into current events, we see that right now with COVID. Like the people who get the sickest and the people who die from COVID are not dying because the virus is taking over the body. For the most part, by the time you're deathly ill, in those fraction of patients that become deathly ill, the virus is gone. And it's the damage that the immune response has done to your body trying to fight that virus that actually is so detrimental. So it's really, the way I think about it, it's really two sides of one coin. You have the microbe attacking the host, and the host trying to attack the microbe. And sometimes the tools to attack the microbe are very specific and really win that battle, but sometimes they're non-specific, and there's a lot of damage that happens. And I got really interested in that kind of two sides of that coin, and I started looking around for opportunities in what we call innate immunity, which is the part of our immune system, our immune response, I should say, that doesn't involve antibodies and T cell receptors, so-called recombinant antigen receptors, but instead relies on receptors that are hard coded into our genome. So that's sort of how I got into immunology. It was a very circuitous sort of route, and I started looking for opportunities in innate immunity at that time. And it was a budding field because it was I graduated my PhD in 1996, and in 1989, I think, a very famous immunologist from Yale, Charlie Janeway, published a very famous paper now in Cold Spring Harbor Journal about how immunologists and immunology as a field was ignoring the innate immune response, and how this innate immune response and innate triggers were critical for driving an antibody response, so critical for making a good vaccine, and that we needed to try and understand how that worked. We used these things called adjuvants, but we didn't really understand how they worked. That was his classic example, and he theorized on how this might occur. And I started reading his work and that paper and follow-up papers as him and other people started to try and tackle this important question. It was really fascinating. About the same time, and inspired by his thoughts, a couple groups in Europe started working on Drosophila immune responses as a model system to study innate immunity. So Drosophila and insects in generally don't have what the opposite of an innate immune response is, something we call the acquired immune response. So insects generally don't have an acquired immune response. There's some exceptions where some invertebrates have some pretty funky things, but no insects have B cells and T cells and RAG genes and somatic recombination like we do, and they generally don't rely on recombinant antigen receptors to detect pathogens. So it was this, but they do have an immune response. It was quite clear, and the critical work was actually done in the 1970s by a guy in Stockholm named Hans Boman, and he noticed that, like, if you infect the fly a little bit and then come back a couple of days later and infect it with a lot of bacteria that would normally kill it, the flies were protected. And he noticed this protection, and from that he went on to purify and isolate and clone a group of small proteins called antimicrobial peptides.
Interesting. And I think 2011 Nobel Prize in medicine I think went to innate immunology. I think 2011 Nobel Prize.
Yep, that's right. So that was and one of the winners of that prize was Jules Hoffman, and he is the other pioneers in this field. Like, so Hans really came first, and I always thought Hans should be included in the Nobel Prize, but he was not. And he discovered these antimicrobial peptides, and then Jules and in France and a number of labs in Sweden, all of whom had trained in Hans Boman's group. And interestingly, Jules had also done like a sabbatical in Hans's group. I think his wife originally went to Hans's group to work for a year, and then Jules, I think, came for the summer and got really inspired and went back to Strasbourg and refocused his group on the same area. Hans had discovered these peptides, and going back to like what I studied in graduate school, these antimicrobial peptide genes, so they're encoded by genes. In a healthy animal, they're basically not transcribed. They're basically almost completely off. And then if you infect the fly or immune challenge the fly, those genes get induced 100, 1,000-fold. It's really a massive induction of those genes. So I was interested in starting my postdoc, and Jules Hoffman, who won the Nobel Prize, and a number of labs, including Hans Boman and Håkan Steiner and Elva Engström in Sweden, where everybody was kind of fascinated by this question: how do these genes get turned on by the immune response? And Jules went on to do critical work showing that the toll pathway was really important for part of that response. The work, some of the key work in his lab, I want to just make sure I mentioned some of the key people who did the work, but Bruno Lemaitre, who's in Lausanne now in Switzerland, has his own group. He was really leading those studies within Jules's larger laboratory. And so they discovered the toll pathway was important for part of the story, and Bruno and Jules also discovered a separate pathway called the IMD pathway that is also very important. And those two pathways have some important similarities. In particular, they both activate a certain kind of transcription factor called NF-kappa B, which is also really important in the human and mouse and the mammalian innate immune response. NF-κB is kind of one of the critical transcription factors of innate immunity. So you have this similarity, and you can study flies and maybe learn something about mammals. And in fact, Jules's group showed the toll pathway was important, and shortly thereafter, Charlie's group with Ruslan Medvedov and Bruce Beutler's group, and Bruce was one of the other Nobel Prize winners, showed that the human TLRs were playing a very similar role in activating the innate immune response in mouse and human cells. The third person on that, so Jules, so the Nobel Prize is funny, right? They can give it only to 3 people, and they can split it different ways. They can do like 1/3, 1/3, 1/3, or they can actually give it to 2 different discoveries, and like 2 people get half, and the third person gets it. So that year, the prize was split in half, and Ralph Steinman, who died like days before the prize was announced because he had cancer. I think he had pancreatic cancer, but I don't remember exactly. He was battling cancer for a decade. And it was very unusual because you can't win when you're deceased. That's another rule. But they announced it, and then I think the next day he died. So he still won, even though he couldn't attend the ceremony. He went for the discovery of dendritic cells, which are a certain kind of cell in the immune system, kind of white blood cell that's really, really important, part of the innate immune response that talks to the adaptive immune response. So there's important communication that has to happen for, say, a B cell to get activated and make those antibodies that we all want to protect us from, say, a coronavirus. So he won half the prize for the discovery of dendritic cells, and then Jules Hoffman and Bruce Beutler won half the prize.
Very, very interesting, I think.
Yeah. But this guy Beaumont, he gets the short shift. His stuff was critical to foundation for these discoveries. Charlie Janeway, who also died young from cancer, also his thinking about the whole problem was really important. And Hans's discovery of the antimicrobial peptides is central to the whole tale. So it really, it took, I think Hans's first paper on this was 1972, if I remember correctly. And then the first toll paper about toll and immunity in Drosophila was 1990, is it 96? Might have been 96, 97. So over 20 years to go from there, from that initial discovery to Nobel Prize winning work. And then there's this whole other story of where toll was originally discovered, which is fascinating. I could tell you about it if you want to hear about it. But it was discovered its initial discovery had nothing to do with immunology, and its initial discovery was because it's involved in the early embryonic development, the early embryonic patterning of the Drosophila embryo. A totally distinct role for this protein and the whole pathway. So there's also some serendipity.
Very, very interesting. And now I think we will change some gears and really get into a profession. I think, what is the least and most exciting part about your profession?
Oh, the least exciting?
Yeah, most exciting.
The least exciting are all the boring meetings I have to attend. I kid you not.
Okay.
So as a faculty member now, finished graduate school, did a postdoc, got a job as a faculty member. I run a research lab, right? So I have to, I manage what goes on in the lab. So I have 6, 7, 8 people in the lab. I talk to them a lot. We talk about experiments and results, and that's all really fun. New discoveries, that's the best thing. When somebody walks into my office, or nowadays they Zoom with me because we're not doing anything in person, and they have and they show me new data, like here's a graph, here's a picture, here's a big Excel spreadsheet, look at this. And there's some new discovery, and those are the best days. Those are the best days. And it's not every day, right? It's not every day. It's sometimes not even once a month. Though that's fabulous. But other things I do, I have to, I'm involved in teaching, and I'm involved in helping run our graduate program, and all that stuff takes a lot of coordination and communication. So there's a lot of time spent in meetings with my colleagues, and sometimes they're a little dull. I don't too many meetings.
I can feel you. I can feel you. But still, I think you have spent many years in this immunology field. Uh, and what has been your toughest challenge till now? That make you toil very hard.
Well, I think the toughest challenges stay the same. So the lab and the work in the lab gets performed by graduate students and postdocs. I sometimes, like right now, I have a research associate lab manager who helps with some of the stuff. But the team is always changing. Students graduate and they go on to do other things with their life. Postdocs want to get quote unquote real jobs where they're going to earn a better living and be in charge of their own science, or maybe they leave science and do something else, whatever they want to do. So it's always constant. So managing the personnel is really, that's a constant challenge that I toil on all the time. And everybody who works in the lab is different. You know, every single person it's kind of amazing, people, right? Everybody's different, right? And you have
everyone has a different approach to the thinking,
a different approach and a different personality, and I have to adjust how I interact with everybody to try and help them get the most out of their experience in my lab, because all those positions as graduate students or postdocs, they're training positions. So I'm supposed to be teaching them about how to be a scientist. So I try and adjust my approach, and I try, you try and keep track of who's going when, and when you're going to hire somebody new. And, and the added complication that I sleep over sometimes is the money is not constant, right? So a big part of my job is writing grants. So I'm constantly writing grants. I'm constantly chasing money to do our experiments. Where does the money go? The big thing the money goes to is to pay the salaries of myself and everybody in the lab. And of course, all the stuff that we buy is very expensive as well. Reagents, enzymes, chemicals, plasticware. The animals cost money. So I'm constantly writing grants. I submitted 2 grants in the last 2 weeks. For example, I'll submit one next month, and I have plans to submit a new grant in June, and another one in July, and hopefully one in September. That's my year, writing grants. Just so you know, I'm not exaggerating. I'm writing grants all the time, and it's a little relentless. So that's not always the most pleasant thing about my job, but it's critical, because there's no money, then the experiments come to a halt, and then the data comes to a halt, and then the whole operation falls apart, and I'll have to find something new to do with my life. So those are some of the big challenges that I have is balancing those things, and trying to optimize everybody's experience in my lab, as well as the productivity coming out of my lab. And I'm not always successful. Sometimes we do better than others. Sometimes some projects take a long time to come to fruition, and it can be quite frustrating, but we get there.
So my question is, I think that a lot of people come with certain preconceived notions when they are, entering the research lab, especially graduate assistants, or certain people would have assumed certain things about your career, like what would a person running a research lab would do. So what are some of the things which have been like sort of myths or just assumptions and not completely true that people have, let's say, talked about to you?
Yeah, that's a good question. I think there's a lot of assumptions. So we can talk about assumptions people make about what it's like to work in a lab, especially if you're a graduate student, a postdoc, and you're actively in the lab. And I think a lot of people who are not in science assume it's kind of a sterile environment with not a lot of people interaction. Like here, well, I'm a people person. I don't think lab work is for me. I'd rather go be a doctor. And I think that's just totally wrong, because the laboratory is a very social environment. Everybody in my lab works on things that are related to each other, and they better be talking to each other all the time about their science, because what person number 1 is doing relates to what person number 2 is doing. Maybe if they talked a little bit, they'd have some brilliant idea for an experiment they could do together that would just blow the whole thing open. So I actively try and encourage those interactions, and 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 we also just socialize just for fun, just to keep the environment fun. And our laboratories at my university are like a big open space. So I have 3 bays, but there's 26 bays in this giant room. So we're also talking to the people working in the labs next door. When it's not the middle of a pandemic, we're lunching, we're having lunch together, we're having joint group meetings together. There's a lot of talk, a lot of social interaction. Some of it's purely social, but a lot of it like sort of science social, like talking about your project, talking about your experiments, going to talk to this guy down the hall or upstairs about how they do this assay. So it's actually really interactive, people facing occupation. So that's one misconception. Another misconception, when I tell people I'm a professor, they ask me what I teach, and I tell them with a straight face, as little as possible. And they look at me and they go, what? Huh? And I laugh. I go, well, my real job is research. Like, I'm running a research lab. That's what I do. I do some teaching, but oh, okay. Well, but what do you teach then? I teach immunology. Like, they can't, like, if they hear a professor, they're just assuming I'm spending most of my day in the classroom. And you can't convince some people of otherwise. That's not what professors at research institutions do with most of their time. Those are the 2 big assumptions and misconceptions that I see. A little bit trivial, but that's what I see.
You belong to American education system. I know you grew through that, and what if I give you a chance and return to that and change some parts of that education system that you don't like, and what is going wrong in the overall education system in the world?
I'm not an expert on education. I mean, I'm an educator, but I'm not an academic in the education field. So I feel like I'm maybe getting out of my area expertise here. But from what I see, I would like to see at the college level, in the life sciences, because that's what I know, less emphasis on memorizing facts and more focus on the experimental approaches that provided the understanding. I think it's more interesting. It's more engaging to try and understand how people actually did these experiments. How do we know that genes are encoded in DNA, right? Just very, very basic. And if you graduate in life science, you should know the classic experiment done by those guys at Rockefeller, Avery, McCarty, McLeod, I think were the authors. I might have the order wrong, where they showed that DNA was responsible for transforming one strain of smooth pneumococcus into a rough strain of pneumococcus. And it turns out the rough strain's virulent, I think, and the smooth strain's not. I don't remember exactly. And so they showed that they could, this transforming principle, they purified it, and it was DNA. And that was the first experiment really to show that DNA contained genetic material. And it took that paper was published, and it didn't, like, the whole world didn't change. It took a while before people realized they brought it. Anyways, I digress. I'd like to see more emphasis, that's my answer to your question, on the experimental basis of our knowledge and less emphasis on memorizing facts. If you go to a big university and you rely on, the classes rely on multiple choice tests, it's hard to get away from just memorizing. You can with well-written test questions. I'm not good at writing those questions, but I definitely don't. In graduate school, we almost never use multiple choice questions.
I have the same problem in biology. The taxonomy in the animal kingdom, plant kingdom, and you have to remember all those names.
I look it up. I don't know any of that stuff. I forgot it the day after the exam when I was in college. Yeah, like that's like my brain's too small. Like I have no room for taxonomy. I mean, it's in Wikipedia. Just Google it. I mean, I think it's stupid to ask people to memorize that kind of stuff.
Yeah, what do you feel? I think how to develop scientific thinking at a very young age. It is very important if you go in research or I think any background. I think scientific thinking is very important. Whatever background you choose, how to think exactly is very important. And what do you think on that?
It is. It is. Well, if you're interested in science, I would encourage anybody of any age and parents of young kids to experiment, like actually like try and figure things out. And experiments can be, simple, like taking apart a toy or a bicycle or something like that, especially if you're kind of more into engineering, like taking things apart, putting them back together, um, exploring the natural world. If you're into the sort of botany, zoology type things, score how many birds come to your bird feeder or what kind of birds show up. There's a lot of things you can do as a citizen scientist, and it doesn't need to be particularly fancy or sophisticated to start doing interesting things. I send a bird feeder because I got a bird feeder right up there. There's some birds, and it's middle of winter, but there's some birds over there in the tree. You could change the food. Does it change the kind of birds that show up? There's things there's lots of little things you can do. All sorts of ages. You don't have to be a PhD scientist to ask interesting questions and try and explore them in the world around us. Because I just think that's way more interesting than trying to soak in facts, specific knowledge. If something gets you, if you're interested in something and you start exploring it, the things that underlie it that we already know, you'll pick up and you'll absorb and you'll know them without even trying. And the fun part is the exploring and I still love discovery. Like I told you, I got into this because I wanted to discover new things, and that's still my favorite day when somebody in the lab has some new results and they come show me, and it's just, even if my original idea was all wrong, it's still a great day. Look at that hypothesis was totally wrong. We had it backwards, whatever. Nobody knew this 30 minutes ago, and now we know this. That's the best feeling.
I have one question. So, if you had to take like a futuristic view of the profession, that is like biomedical research, how do you think, say, even 10 years or 30 years down the line, would the field evolve? So, what is your perspective on how the future of biomedical
And if
research?
you would have asked me that 20 years ago, I would have gotten it totally wrong. So I'm not sure I'm good at prognosticating. The big change that's happened in the last 10 or so years is the ability to sequence things, massive amounts of sequencing data through totally new technology to sequence. That's totally changed the way we do a lot of things in the laboratory. It affects the life sciences, but it affects all of us too, to some degree. This is how these new variants of the coronavirus were discovered by sequencing them, which 20 years ago would have taken months and months and months, and now it takes a couple of days, a couple hours even, if you have a pipeline set up. So what's the next big revolution? I think sequencing is still going to be huge. There's going to be more advances in sequencing, and you're going to be able to read more sequences more fast and from more smaller samples and read longer reads. I think that technology is continuing to advance at breakneck speeds, and it's going to continue to change the way we think and do experiments in the biomedical sciences. Another big change that's happening, so this isn't even prognostication, this is happening, has to do with CRISPR, right? So you guys probably heard about CRISPR, and you can use CRISPR to engineer
genetics.
We call in the DNA. You can we call that mutants. You can make designer mutants.
If I remember, clustered, regular, interspaced, palindromic repeats, right?
So your memory.
I did a research paper. I did a paper on that.
So that technology is rapidly advancing too. That's there's the original CRISPR system, which is CRISPR-Cas9, and there's a a bunch of other ones that are also derived from different bacteria that can be sort of utilized in similar manners that have different properties. So that's really going to change. We're going to be able to safely manipulate human embryos if we so want to. I mean, that's an ethically fraught thing in the very near future. And we're going to have, as a society, we're going to have to figure out how to regulate that properly. And in the laboratory, it means we can, in our favorite genetic model systems, do amazing things to try and understand how genes and proteins work. But it also, like, another revolution there is coming, because you can use CRISPR to genetically engineer organisms that aren't typically used in the laboratory, or were not typically used in the laboratory at least 10 years ago, because there was no good way to manipulate their genome. But now you can do CRISPR in almost any genome you want to, if that's, if you want to study ants and how they, ants are social insects, right? They form these colonies. There's lots of cool questions about ants. I think people are doing CRISPR on ants, and you can CRISPR different bacteria genomes that you couldn't use. So CRISPR is going to be amazing, and it's going to open up new areas of biology to study at the molecular and genetic level that weren't available. It's going to revolutionize medicine. Not just in embryos, because you can also take cells out of somebody's body, do something with CRISPR to change their cells and put them back. There's already therapies like that.
We have ethical questions to answer over there, I think, many.
Mm-hmm. Yep. I mean, especially embryo manipulation is particularly ethically fraught.
I think it would change the whole human gene pool, I think, in the coming years. Mm-hmm. And it is irreversible change, I think. What I think, it is irreversible change in a human gene pool, and it is pretty much.
So that's the question. If you're manipulating the germline, right? So you're doing it in embryos. It's absolutely true. If you're like programming a T cell to attack a cancer cell and then putting it back in your body, that's not changing the germline. So that's not irreversible. That's actually quite temporary. So it depends how you marshal these tools. So I think the embryo question is really the key ethical thing that we all need to think about. I don't know
how CRISPR is involved, can be involved in immunology too. How we can write. I saw I think in rats like HIV virus, about HIV virus. And we can be immune to HIV. I think. I'm not in.
I know what you're talking about. So, like, the problem with HIV, right? So the problem with HIV is you can go on HAART, right? A highly effective antiviral retroviral therapy, which is like I think it's a triple drug cocktail, right? You have a reverse transcriptase inhibitor, a protease inhibitor, and a fusion inhibitor, and you can get the patient's level of HIV in the blood down to almost to indetectable levels. But then if you take those patients off the drugs, inevitably, in most patients, like 99% of the patients, the HIV comes back when you take them off the drug, because the HIV is hiding somewhere, right? HIV is a retrovirus, so it's RNA, and when it gets into the cell, it gets reverse transcribed into DNA and inserts into your genome. So it's literally hiding in some cells somewhere in your body in the DNA, and then you take away the drugs, it slowly starts to come back. And so you have these latent infections somewhere, and the problem is how do you get rid of those to really cure somebody, to really get rid of the virus and the viral genome, so you can take them off the drugs. And because the drugs have side effects, right? I mean, people are on those drugs for the rest of their life, but they're hard on the body. So there are definitely people trying to use CRISPR to attack those latent pro-viral genomes that are hiding in cells. There's a lot of interesting questions, because, like, what cells are they in? Can the immune response tackle? There's a lot of CRISPR is one of many things that are going on in that area. That's a very interesting and important area of study in HIV right now, trying to get those reservoirs of HIV. But if you knew where they were and you knew how to deliver CRISPR there, you could design CRISPR to go cut out and remove that HIV and maybe get rid of it. That's sort of the idea.
I would like to ask, like you had taken a lot of detours in your career, like you experimented a lot. So if you had to give anybody, say, an advice to design their own life or just like design their career, what advice would you give to anybody in that regard? Because you have experimented a lot, and I think you would have something like good and a great, unique advice for anybody in any
Uh,
career in that sense.
yeah, well, I don't know about any career, but so I'll answer mine in sort of, I'll narrow my scope to a career in life sciences, especially in academia. My advice is to find projects that you're really passionate about. You know, everybody's different. Some people want to cure important diseases, whether it be AIDS or COVID-19 or cancer. Other people just want to understand how the natural world works. Other people really like sort of the idea of engineering and tinkering with things and figure out how things work and make new things that work better or to their design. Find what it is that you really like to do. Find a project that really excites you and be passionate about it. I think that's the first thing. And remember, you know, when you're on a project, it's not your whole life. You got to get the project to some sort of endpoint. You don't have to, but it's a linear way through the science careers. Get that to some endpoint, and then you can move on to some other project if you get sort of tired of that project. You know, I did that at graduate school. I had enough of that project and that topic by the time I finished graduate school, and I took a left turn and did something else. I needed something fresh and new, and that's fine. I was excited about that project at the beginning, and then I was ready for a new topic. And then my other advice is find people that you like to work with and for, your lab mates, your advisors, your colleagues. These people really make your day-to-day better or worse. And it's not just that you like them and you want to go out and have a beer with them after work, which is nice sometimes, but it's not necessary. You know, some people have families and they have to get home, but that they're good colleagues. So they're respectful, they're thoughtful. When you ask them for help, they help you, whether it's, you know, with some reagent you might need or teaching them how to do some sort of protocol. But they're genuinely helpful and collaborative and interactive, and that they're smart. You want to be around smart people, and smart people don't only live at the fanciest schools. There's smart people all over the globe, and you want to find them and bring them together, because they'll make you smarter too, because you get them thinking about your problem. They might have some brilliant ideas. So projects you enjoy, move on to new projects when you've had enough. People you enjoy, smart people you enjoy interacting with, and always spend some part of your time just thinking about your project and what you're working on, thinking, reading the literature, going to seminars. When things are looking bleak, it's good to take a few days off and regroup and think about it, and maybe go to a conference or a bunch of seminars and learn some new things and get inspired to do something new. Life's too short to be miserable.
So on this high note, we can end this podcast. And thank you very much, Dr. Neal Silverman, for coming on this podcast. It was very knowledgeable. I really enjoyed it very much, and we're considering immunology for research. Thank you very much.
Good luck to you finishing up high school, and I hope you find a spot in a nice university that you love, and you have some great instructors who focus on experiments and not memorization. And good luck on the literature and the advertising too. It was nice to talk to you both. Nice to meet with you. Let me know when the podcast is done so I can check it out.
Definitely, sir. Definitely.
Thank
Thank
you.
you. Bye.
My pleasure. Bye-bye.
Bye.